Good afternoon, everyone. Thank you for joining this session. My name is Mohammad Muntasir Rahman, and I am a postdoctoral associate at Mississippi State University. I am working under Dr. Taibi's lab here. First of all, I would like to extend my heartfelt gratitude to the conference organizer for giving me this opportunity to present this research work. Today, I am going to present my paper titled Extracting Cardiovascular-Induced Chest Vibration from Ordinary Chest Videos, a Comparative Study. This study was conducted as part of our ongoing research to develop non-invasive and accessible method for monitoring cardiac health. So here is the introduction and motivation. So cardiovascular diseases are the leading causes of death worldwide, which is responsible for approximately 17.9 million deaths yearly. Beyond the human toll, the economic burden of this disease is projected to reach 47 trillion USD by 2030. So early detection and effective monitoring are crucial for reducing this impact. Existing diagnostic tool like electrocardiography focuses on electrical activity, but the mechanical vibration of the chest can also provide complementary insight into cardiac function. So SCG or seismocardiography captures this mechanical vibration of the chest, which is caused by heart activity. However, traditional SCG relies on chest-mounted accelerometer, which are sometimes inconvenient and costly. To overcome these challenges, we explored SCG extraction using computer vision-based method, which can offer a non-contact and accessible alternatives. So previously, we developed vision-based SCG extraction method using three computer vision-based methods: Lucas Kennedy optical flow-based SCG, template tracking-based SCG, and Garner-Fernbeck optical flow-based SCG. These methods were successfully published in journals such as Scientific Report and also accepted in NPJ Cardiovascular Health, and also we presented at the IEEE Body Sensor Network conferences, that validating our potential for non-contact SCG extraction. So our objective has three main objectives. So first of all, to evaluate the feasibility of using computer vision to extract XCG signal from video. And second, compare the performance of these three vision-based methods using Lucas Kennedy optical flow, template tracking, and Garner-Fernbeck optical flow. Finally, we will validate the extracted signal by comparing them with the gold standard accelerometer data. We will use metric like correlation, mean squared error, and root mean squared error. Here is the materials and method of our work. So now let's discuss how we conduct this study. For this study, we recruited 13 healthy participants with no cardiovascular disease history. So if you see in the figure, you can see the data acquisition and sensor placement setup. In our experiment, when we recorded the data, all subjects were positioned in a supine position, and an accelerometer were attached to the left sternal border near the fourth costal notch. To facilitate the motion tracking, we attached a QR code on top of the QR code, on top of the accelerometer. Data was collected during two breath hold sessions, at the end of exhalation and at the end of inhalation. Accelerometer was used to record SCG signal, and we used an iPhone 13 Pro to capture the video at 60 frames per second. Both accelerometer and video recording were synchronized using a microphone. So here we describe our vision-based SCG extraction method. In step 1 is the QR code sticker detection. For this purpose, we trained a yellow version 7 model on a custom dataset to identify the QR code in the first frame of the video. In second step, we track those QR code using computer vision based method. So when we detect the location of the QR code in the first frame of the video, then we apply Lucas Kennedy template tracking or Gunner framework motion tracking to track those QR location in the subsequent frame. And then we get a displacement signal. Then we calculate the SCG signal as the acceleration, which is the second derivative of the displacement signal. If you see the figure 2, it shows the vision-based SCG extraction process. So here we use the YOLO version 7 only for the detecting the QR code in the first frame. And then we apply the Lucas-Kennedy template tracking and front-back optical flow method to track those QR code in the subsequent frame. So after getting the SCG signal, we applied some post-processing step. For signal denoising, we applied a bandpass filter with a cutoff frequency 1 to 30 Hz to remove the low frequency noise and respiration effect. Then we applied, then we resampled the signal from 60 Hz to 5000 Hz to match the acceleration's sampling rate. Finally, we normalized the signal to improve the comparability. Let's examine the extracted signal from a subject. In this figure, shows the comparison of vision-based SCG signal with a gold standard accelerometer signal. Here you can see the ECG signal is here, and the second row shows the accelerometer signal. And left plot shows the SCG signal is in the right to left direction, and right plot shows the SCG signal in head to foot direction. Visual inspection confirms that the vision-based SCG signal is very close to accelerometer signal, with resemblance between the signal extracted by 3 vision-based method and gold standard accelerometer signal. This indicates that our method successfully captured cardiovascular-induced chest vibration. Now, to quantify the similarity between the signal, we calculated the mean squared error, root mean squared error between the vision-based SCG signal and the accelerometer signal. Here, the figure shows the bar plot of the mean squared error of all methods. For the head-to-foot direction, MSE ranged between 0.14 to 0.93, template tracking ranged between 0.12 to 0.94, and Gunner Funbeck ranges between 0.2 to 1.07. For the right-to-left direction, MSE was generally higher, ranging between Lucas Kennedy 0.5 to 1.46, template tracking 0.58 to 1.47, and where the Gunner front brake 0.67 to 1.56. Let's look at the root mean squared error. If we see the figure, we can see that the RSME value also followed the same trend, with values being lower at head-to-foot direction across all methods. Head-to-foot RMSE range from 0.3 to 0.35 to 0.96 for the Lucas Kennedy and template tracking methods, while the Funbeck method shows slightly higher values. Right to left RMSE ranges from 0.70 to 1.25. The lower MSE and RMSE value in the head-to-foot direction highlighted that this axis is better suited for vision-based SCG. This is likely due to the strong cardiovascular-induced motion along this axis. Among the 3 methods, Lucas Kennedy and template tracking achieved comparable performance with lower MSE and RMSE values. In addition to MSE and RMSE, we evaluated the correlation between the vision-based method and the gold standard accelerometer signal. In our case, the correlation value ranges between -1 to 1. For the head-to-foot direction, Lucas Kennedy achieved an average correlation of 0.82 plus minus 0.09. Template tracking achieved 0.83 plus minus 0.1 and gunner front back achieved 0.76 plus minus 0.11. For the right to left direction, correlation were lower. Lucas Kennedy and template tracking achieved around 0.5 to plus minus 0.12, while the gunner frame brake achieved 0.44 to 0.13. Here we can see that the Lucas Kennedy and template tracking consistently achieved stronger correlation, particularly in head-to-foot direction. So to conclude, here are the key findings of our study. Lucas Kennedy and template tracking method demonstrate superior performance in terms of accuracy and correlation. Vision-based SCG signal is especially effective in the head-to-foot direction, where the vibrations are more pronounced and easier to track. And lastly, this approach offers a promising and non-contact alternative for cardiac monitoring, and which might be more accessible and comfortable for patients. So this is our future work. In future, we are planning to expand the study to a larger and more diverse populations. And we will integrate this method with machine learning to further improve the accuracy. We'll also focus on optimizing environmental factors like lighting and camera environment setting for the real-world applications. So here is the reference of our work. Thank you, everyone. I'm happy to answer any question.
Thank you. Any questions?
Yeah, could you please go to the page 6 where you describe the signal noising?
Page 6.
Yeah, are those operations sequential? The band pass filter, resample, normalize the signal?
Yeah, it is sequential.
So after the band pass, I think you say added here 1 to 30 hertz. So after the band pass, I think there will be no signal that has 60 hertz. So you are resampling 60 hertz to 5000 hertz. How?
Band pass signal, we are actually band passing the frequency 1 to 30 hertz, but we recorded the data with 60 frame.
Yeah. After the band pass,
Yeah.
the frequency will be eliminated, right? Those are not in between 1 and 130 hertz.
Yes, we applied first bandpass signal to filter the signal between 1 to 30 hertz because we recorded the data with 60 frame per second. So at most we can gather 30 hertz. So we filtered out the signal between 1 to 30 hertz. And then since our accelerometer data was captured with 5000 hertz, that's why we resampled it to 5000 hertz to match with the ground truth, so that we can compare it with the accelerometer.
After the band pass, I mean, if the
Okay, here
operation are
maybe
sequential,
I wrote here incorrectly, but we are first applying
yeah, that's maybe,
bandpass
yeah,
filter and then resample it to 5000 to match with the accelerometer.
yeah, that's I am telling you that after the band pass, there will be no signal other than
Yeah,
between 1 and
right.
30
But
hertz.
we are trying to resample it to match with the accelerometer signal, since it was recorded with 5000 hertz.
Okay, thank you.
Okay, we have another comment from Amir. I think it's related to the same thing. Would you like to ask, or were you answered already through this discussion, Amir? Or was your question answered?
Yes. Thank you very much. So yeah.
Can you go a slide or two after? when you
Like
show, you should go a little bit. I think you had the waveforms. You were comparing the waveforms.
waveforms here.
Yes, this one.
Yeah,
You see, when I look at the accelerometer versus let's look at the right column.
yeah.
And the second waveform is the
Second
accelerometer.
waveform. Yeah.
The
The
waveform
second
after
waveform
that,
comes from the accelerometer.
yes, the waveform after that appears to have a little bit of high frequencies. Are these noise or are these things that were not detected by the accelerometer in your opinion?
It actually a noise because when we capture those SCG signal from the vision-based method, we always see some high frequency noise. Those high frequency noise clearly seen in the right to left direction signal, because we found that since the right to left directions movement is very subtle, so in that case we are actually getting more noises than the head to foot direction.
So if you feel it is noise, do you think we should filter it because you think it's noise?
We're trying to filter it and we still trying to find out a way how to get rid of these
So what
noises
frequency is
because
that? Do you know?
sorry.
This noise, what frequency? If you don't know, it's okay. I just was wondering. And you mentioned something about the head to foot is is the strongest. I
Yes.
believe that I believe
Yes,
in that.
if you see this graph and if you compare the accelerometer signal with the vision-based signal, this right portion shows the head-to-foot direction signal. And if you see this signal is very similar, but if you compare to right to left direction signal, then you see we have some noises here. That's why we are saying that
So you
head to foot is more pronounced because the
yeah,
heart vibration is more
there
pronounced
is
in
no
that direction.
the vertical axis is not labeled, so it's difficult to know which one is louder. But that's why I'm asking. Now I have a question. You measured only two components, left to right and head to foot,
Yes.
and the third component you couldn't measure. Is
No,
that right?
because from the vision-based method, we only capture left to right and head to foot. We cannot detect dorsoventral direction. So in
Okay.
that case, we need actually 3D data. We only have 2D data, so we cannot capture it.
Okay. Okay. Thank you.
You are welcome.